In livestock, the genetic diversity of livestock species permits animal husbandry in a range of environments and with a range of different objectives. It provides the raw material for selective breeding programs and allows livestock populations to adapt as environmental conditions change. 29. Livestock biodiversity can be lost as a result of breed extinctions and other forms of genetic erosion. As of June 2014, among the 8,774 breeds recorded in the Domestic Animal Diversity Information System, DATIS, operated by the Food and Agriculture Organization of the United Nations, FAO, 17% were classified as being at risk of extinction and 7% already extinct. 29. There is now a global plan of action for animal genetic resources that was developed under the auspices of the Commission on Genetic Resources for Food and Agriculture in 2007, that provides a framework and guidelines for the management of animal genetic resources. Awareness of the importance of maintaining animal genetic resources has increased over time. FAO has published 2 reports on the state of the world's animal genetic resources for food and agriculture, which cover detailed analyses of our global livestock diversity and ability to manage and conserve them. Viral implications. High genetic diversity in viruses must be considered when designing vaccinations. High genetic diversity results in difficulty in designing targeted vaccines, and allows for viruses to quickly evolve to resist vaccination lethality. For example, malaria vaccinations are impacted by high levels of genetic diversity in the protein antigens. 30. In addition, HIV when genetic diversity limits the use of currently available viral load and resistance tests. 31. Coronavirus populations have considerable evolutionary diversity due to mutation and homologous recombination. 32. For example, the sequencing of 86 SARS-CoV-2 coronavirus samples obtained from infected patients revealed 93 mutations indicating the presence of considerable genetic diversity. 33. Replication of the coronavirus RNA genome is catalyzed by an RNA-dependent RNA polymerase. During replication, this polymerase may undergo template switching, a form of homologous recombination. 34. This process, which also generates genetic diversity, appears to be an adaptation for coping with RNA genome damage. 35. Coping with low genetic diversity. A Tanzanian cheetah. Natural. Photomontage of planktonic organisms. The natural world has several ways of preserving or increasing genetic diversity. Among oceanic plankton, viruses aid in the genetic shifting process. Ocean viruses, which infect the plankton, carry genes of other organisms in addition to their own. When a virus containing the genes of one cell infects another, the genetic makeup of the latter changes. This constant shift of genetic makeup helps to maintain a healthy population of plankton despite complex and unpredictable environmental changes. 36. Cheetahs are a threatened species. Low genetic diversity resulting poor sperm quality has made breeding and survivorship difficult for cheetahs. Moreover, only about 5% of cheetahs survive to adulthood. 37. However, it has been recently discovered that female cheetahs can mate with more than one male per litter of cubs. They undergo induced ovulation, which means that a new egg is produced every time a female mates. By mating with multiple males, the mother increases the genetic diversity within a single litter of cubs. 38. Evolutionary biology portal. Human intervention. Attempts to increase the viability of a species by increasing genetic diversity is called genetic rescue. For example, 8 panthers from Texas were introduced to the Florida panther population, which was declining and suffering from inbreeding depression. Genetic variation was thus increased and resulted in a significant increase in population growth of the Florida panther. 39. Creating or maintaining high genetic diversity is an important consideration in species rescue efforts, in order to ensure the longevity of a population. Genetic diversity of a population can be assessed by some simple measures. Gene diversity is the proportion of polymorphic loci across the genome. Heterozygosity is the fraction of individuals in a population that are heterozygous for a particular locus. Alleles per locus is also used to demonstrate variability. Nucleotide diversity is the extent of nucleotide polymorphisms within a population, and is commonly measured through molecular markers such as micro and mini-satellite sequences, mitochondrial DNA, 40, and single nucleotide polymorphisms, SNPs. Furthermore, stochastic simulation software is commonly used to predict the future of a population given measures such as allele frequency and population size. 41. Genetic diversity can also be measured. The various recorded ways of measuring genetic diversity include 42. Species richness is a measure of the number of species. Species abundance, a relative measure of the abundance of species. Species density, and evaluation of the total number of species per unit area. Biodiversity. Inbreeding depression. Inbreeding depression is the reduced biological fitness that has the potential to result from inbreeding, the breeding of related individuals. The loss of genetic diversity that is seen due to inbreeding results from small population size. 2. Biological fitness refers to an organism's ability to survive and perpetuate its genetic material. Inbreeding depression is often the result of a population bottleneck. In general, the higher the genetic variation or gene pool within a breeding population, the less likely it is to suffer from inbreeding depression, though inbreeding and outbreeding depression can simultaneously occur. Inbreeding depression in Delphinium nelsoni. A overall fitness of progeny cohorts and the bee progeny lifespan were all lower when progeny were the result of crosses with pollen taken close to a receptor plant. 1. Inbreeding depression seems to be present in most groups of organisms, but varies across mating systems. Hermaphroditic species often exhibit lower degrees of inbreeding depression than outcrossing species, as repeated generations of selfing is thought to purge deleterious alleles from populations. For example, the outcrossing nematode roundworm Caenorhabditis riemanni has been demonstrated to suffer severely from inbreeding depression, unlike its hermaphroditic relative C. elegans, which experiences outbreeding depression. 3. Example of inbreeding depression. Mechanisms. Inbreeding, i.e. Mechanisms. Inbreeding, i.e., breeding between closely related individuals, results in more recessive traits manifesting themselves, as the genomes of pair mates are more similar. Recessive traits can only occur in an offspring if present in both parents' genomes. The more genetically similar the parents are, the more often recessive traits appear in their offspring. This normally has a positive effect, as most genes are undergoing purifying selection, the homozygous state is favored. However, for very closely related individuals, there is an increased likelihood of homozygous deleterious genes in the offspring which can result in unfit individuals. 4. For the alleles that confer an advantage in the heterozygous and/or homozygous dominant state, the fitness of the homozygous recessive state may even be 0, meaning sterile or unviable offspring. An example of inbreeding depression is shown in the image. In this case, A is the recessive allele which has negative effects. In order for the A phenotype to become active, the gene must end up as homozygous A because in the genotype A, the A takes dominance over the A and the A does not have any effect. Some recessive genes result in detrimental phenotypes by causing the organism to be less fit to its natural environment. Another mechanism responsible for inbreeding depression is the fitness advantage of heterozygosity, which is known as overdominance. This can lead to reduced fitness of a population with many homozygous genotypes, even if they are not deleterious or recessive. Here, even the dominant alleles result in reduced fitness if present homozygosly, see also hybrid vigor. Overdominance is rare in nature. 4. For practical applications, e.g. in livestock breeding, the former is thought to be more significant. It may yield completely unviable offspring, meaning outright failure of a pedigree, while the latter can only result in relatively reduced fitness. Natural selection. Natural selection cannot effectively remove all deleterious recessive genes from a population for several reasons. First, deleterious genes arise constantly through de novo mutation within a population. Second, most offspring will have some deleterious traits, so few will be more fit for survival than the others. Different deleterious traits are extremely unlikely to equally affect reproduction. An especially disadvantageous recessive trait expressed in a homozygous recessive individual is likely to eliminate itself, naturally limiting the expression of its phenotype. Third, recessive deleterious alleles will be masked by heterozygosity, and so in a dominant recessive trait, heterozygotes will not be selected against. When recessive deleterious alleles occur in the heterozygous state, where their potentially deleterious expression is masked by the corresponding wild-type allele, this masking phenomenon is referred to as complementation, see complementation, genetics. In general, sexual reproduction in eukaryotes has two fundamental aspects, genetic recombination during meiosis, and outcrossing. It has been proposed that these two aspects have two natural selective advantages respectively. A proposed adaptive advantage of meiosis is that it facilitates recombinational repair of DNA damages that are otherwise difficult to repair, see DNA repair as the adaptive advantage of meiosis. A proposed adaptive advantage of outcrossing is complementation, which is the masking of deleterious recessive alleles, 5, 6, see hybrid vigor or heterosis. The selective advantage of complementation may largely account for the avoidance of inbreeding, see kin recognition. However, animals often do not avoid inbreeding. 7. Among animals, inbreeding avoidance is highly variable. 8. Inbreeding avoidance through mate selection appears to evolve only when there is both a risk of inbreeding depression and there also are frequent encounters between sexual partners that are related to each other. 8. Management. Hybridization is a conservation effort as appropriate if the population has lost substantial genetic variation through genetic drift and the detrimental effects of inbreeding depression are apparent, and a similar population should be used. 9. 10. Different populations of the same species have different deleterious traits, and therefore their crossbreeding is less likely to result in homozygosity at most loci in the offspring. This is known as outbreeding enhancement, which can be performed in extreme cases of severe inbreeding, 9. By conservation managers and zoo captive breeders to prevent inbreeding depression. However, intermixing two different populations can give rise to unfit polygenic traits in outbreeding depression, i.e. yielding offspring which lack the genetic adaptations to specific environmental conditions. These, then, will have a lowered fitness than purebred individuals of a particular subspecies that has adapted to its local environment. In humans, inbreeding may have both detrimental and beneficial effects. 11. The biological effects of inbreeding depression in humans can on occasion be confounded by socioeconomic and cultural influences on reproductive behavior. 12. Studies in human populations have shown that age at marriage, duration of marriage, illiteracy, contraceptive use, and reproductive compensation are the major determinants of apparent fertility, even amongst populations with a high proportion of consanguineous unions. 13. However, several small effects on increased mortality, 14. Longer interbirth intervals, 14, and reduced overall productivity, 12, have been noted in certain isolated populations, though other studies show increased fitness of offspring and no effect on lifespan past the second cousin level. 15. Charles Darwin was one of the first scientists to demonstrate the effects of inbreeding depression, through numerous experiments on plants. Darwin's wife, Emma, was his first cousin, and he was concerned about the impact of inbreeding on his 10 children, 3 of whom died at age 10 or younger, 3 others had childless long-term marriages. 16, 17, 18. Humans do not seek to completely minimize inbreeding, but rather to maintain an optimal amount of inbreeding versus outbreeding. Close inbreeding reduces fitness through inbreeding depression, but some inbreeding brings benefits. 19, 20. Indeed, inbreeding increases the speed of selection of beneficial recessive and co-dominant alleles, e.g. those that protect against diseases. 21. In wolves. A small isolated highly inbred population of gray wolves in Isle Royale National Park, Michigan, USA, was considered in 2019 to be at imminent risk of extinction. 22. This gray wolf population had been experiencing severe inbreeding depression primarily due to the homozygous expression of strongly deleterious recessive mutations. 23. Defects arising from severe inbreeding among the wolves included reduced survival and reproduction, malformed vertebrae, syndactyly, probable cataracts, and unusual, rope tail, and anomalous fur phenotypes. 22. A separate small inbred population of gray wolves in Scandinavia was also found to suffer from inbreeding depression due to the homozygous expression of deleterious recessive mutations. 24. Factors reducing inbreeding depression. Whilst inbreeding depression has been found to occur in almost all sufficiently studied species, some taxa, most notably some angiosperms, appear to suffer lower fitness costs than others in inbred populations. 25. Three mechanisms appear to be responsible for this: purging, differences in ploidy, and selection for heterozygosity. 25. It must be cautioned that some studies failing to show an absence of inbreeding depression in certain species can arise from small sample sizes or where the supposedly outbred control group is already suffering inbreeding depression, which frequently occurs in populations that have undergone a recent bottleneck, such as those of the naked mole. Rat. 25-26. Purging selection. Purging selection occurs where the phenotypes of deleterious recessive alleles are exposed through inbreeding, and thus can be selected against. This can lead to such detrimental mutations being removed from the population, and has been demonstrated to occur rapidly where the recessive alleles have a lethal effect. 25. The efficiency of purging will depend on the relationship between the magnitude of the deleterious effect that is unmasked in the homozygotes and the importance of genetic drift, so that purging is weaker for non-lethal than for recessive lethal alleles. 27. For very small populations, drift has a strong influence, which can cause the fixation of sublethal alleles under weak selection. 25. The fixation of a single allele for a specific gene can also reduce fitness where heterozygote advantage was previously present, i.e., where heterozygous individuals have higher fitness than homozygotes of either allele, although this phenomenon seems to make a usually small contribution to inbreeding depression. Although naturally occurring, purging can be important for population survival. Deliberately attempting to purge deleterious mutations from a population is not generally recommended as a technique to improve the fitness of captive-bred animals. 28, 29, 30. In plants, genetic load can be assessed through a test analogous to an inbreeding depression test called an autogamy depression test. Polyploidy. Many angiosperms, flowering plants, can self-fertilize for several generations and suffer little from inbreeding depression. This is very useful for species which disperse widely and can therefore find themselves growing in a novel environment with no conspecifics present. 25. Polyploidy, having more than two paired sets of each chromosome, which is prevalent in angiosperms, ferns and a select few animal taxa, accounts for this. By having several copies of a chromosome, as opposed to two, homozygosity is less likely to occur in inbred offspring. This means that recessive deleterious alleles are not expressed as frequently as with many copies of a chromosome. It is more likely that at least one will contain a functional allele. 25. Selection for heterozygosity. Selection for heterozygosity is rare, as lost loci undergo purifying selection for homozygous loci. 4. Inbreeding depression has also been found to occur more gradually than predicted in some wild populations, such as in the highly inbred population of Scandinavian wolves. This appears to be due to a selection pressure for more heterozygous individuals, which generally are in better condition and so are more likely to become one of the few animals to breed and produce offspring. Genetic purging. Genetic purging is the increased pressure of natural selection against deleterious alleles prompted by inbreeding. 1. Purging occurs because deleterious alleles tend to be recessive, which means that they only express all their harmful effects when they are present in the two copies of the individual, i.e., in homozygosis. During inbreeding, as related individuals mate, they produce offspring that are more likely to be homozygous so that deleterious alleles express all their harmful effects more often, making individuals less fit. Those less fit individuals pass fewer copies of their genes to future generations. In other terms, natural selection purges recessive deleterious alleles in inbred individuals. Purging reduces both the overall number of recessive deleterious alleles and the decline of mean fitness caused by inbreeding, the inbreeding depression for fitness. The term, purge, is sometimes used for selection against deleterious alleles in a general way. It would avoid ambiguity to use purifying selection, in that general context, and to reserve purging to its more strict meaning defined above. The mechanism. Deleterious alleles segregating in populations of diploid organisms have a remarkable trend to be, at least, partially recessive. This means that, when they occur in homozygosis double copies, they reduce fitness by more than twice than when they occur in heterozygosis single copy. In other words, part of their potential deleterious effect is hidden in heterozygosis but expressed in homozygosis, so that selection is more efficient against them when they occur in homozygosis. Since inbreeding increases the probability of being homozygous, it increases the fraction of the potential deleterious effect that is expressed and, therefore, exposed to selection. This causes some increase. Deleterious alleles segregating in populations of diploid organisms have a remarkable trend to be, at least, partially recessive. This means that, when they occur in homozygosis double copies, they reduce fitness by more than twice than when they occur in heterozygosis single copy. In other words, part of their potential deleterious effect is hidden in heterozygosis but expressed in homozygosis, so that selection is more efficient against them when they occur in homozygosis. Since inbreeding increases the probability of being homozygous, it increases the fraction of the potential deleterious effect that is expressed and, therefore, exposed to selection. This causes some increase in the selective pressure against partially recessive deleterious alleles, which is known as purging. Of course, it also causes some reduction in fitness, which is known as inbreeding depression. Purging can reduce the average frequency of deleterious alleles across the genome below the value expected in a non-inbred population during long periods. 2. Which reduces the negative impact of inbreeding on fitness. If inbreeding is due just to random mating in a finite population, due to purging the mean fitness declines less than would be expected just from inbreeding, and, after some initial decline, it can even rebound up to almost its value before inbreeding. Another consequence is the reduction of the so-called inbreeding load. This means that, after purging, further inbreeding is expected to be less harmful. The efficiency of purging is reduced by genetic drift and, therefore, in the long term, purging is less efficient in smaller populations. 1. Purging can be increased if individuals mate with relatives more often than expected by random mating. The joint effect of inbreeding and purging on fitness. Accounting for purging when predicting inbreeding depression is important in evolutionary genetics, because the fitness decline caused by inbreeding can be determinant in the evolution of diploidy, sexual reproduction and other main biological features. It is also important in animal breeding and, of course, in conservation genetics, because inbreeding depression may be a relevant factor determining the extinction risk of endangered populations, and because conservation programs can allow some breeding handling in order to control inbreeding. 3. In brief, due to purging, inbreeding depression does not depend on the standard measure of inbreeding, Wright's inbreeding coefficient F, since this measure only applies to neutral alleles. Instead, fitness decline it depends on the purged inbreeding coefficient, G, which takes into account how deleterious alleles are being purged. Purging reduces inbreeding depression in 2 ways. First, it slows its progress. Second, it reduces the overall inbreeding depression expected in the long term. The slower the progress of inbreeding, the more efficient is purging. A more detailed explanation. In the absence of natural selection, mean fitness would be expected to decline exponentially as inbreeding increases, where inbreeding is measured using Wright's inbreeding coefficient F. 4. The reason why decline is exponential on F instead of linear is just that fitness is usually considered a multiplicative trait. The rate at which fitness declines as F increases, the inbreeding depression rate delta, depends on the frequencies and deleterious effects of the alleles present in the population before inbreeding. The above coefficient F is the standard measure of inbreeding, and gives the probability that, at any given neutral locus, an individual has inherited 2 copies of a same gene of a common ancestor, i.e. the probability of being homozygous, by descent. In simple conditions, F can be easily computed in terms of population size or of genealogical information. F is often denoted using lowercase f but should not be confused with the coancestry coefficient. However, the above prediction for the fitness decline rarely applies, since it was derived assuming no selection, and fitness is precisely the target trait of natural selection. Thus, Wright's inbreeding coefficient F for neutral loci does not apply to deleterious alleles, unless inbreeding increases so fast that the change in gene frequency is governed just by random sampling, i.e., by genetic drift. Therefore, according to the model, the decline of fitness can be predicted using, instead of the standard inbreeding coefficient F, a purged inbreeding coefficient, G, that gives the probability of being homozygous by descent for partially recessive deleterious alleles, taking into account how the frequency is reduced by purging. 1. Due to purging, fitness declines at the same rate delta than in the absence of selection, but as a function of G instead of F. This purged inbreeding coefficient G can also be computed, to a good approximation, using simple expressions in terms of the population size or of the genealogy of individuals, see box 1. However, this requires some information on the magnitude of the deleterious effects that are hidden in the heterozygous condition but become expressed in homozygosis. The larger this magnitude, denoted purging coefficient D, the more efficient is purging. An interesting property of purging is that, during inbreeding, while F increases approaching a final value F equals 1, G can approach a much smaller final value. Hence, it is not just that purging slows the fitness decline, but also that it reduces the overall fitness loss produced by inbreeding in the long term. This is illustrated below for the extreme case of inbreeding depression caused by recessive lethals, which are alleles that cause death before reproduction but only when they occur in homozygosis. Purging is less effective against mildly deleterious alleles than against lethal ones but, in general, the slower is the increase of inbreeding F, the smaller becomes the final value of the purged inbreeding coefficient G and, therefore, the final reduction in fitness. This implies that, if inbreeding progresses slowly enough, no relevant inbreeding depression is expected in the long term. Implies, for example, that the average fitness of a population that has been moderately small for a long time can be very similar to that of a large population with more genetic diversity. In conservation genetics, it would be very useful to ascertain the maximum rate of increase of inbreeding that allows for such efficient purging. Predictive model for the joint effects of inbreeding and purging caused by a reduction in population numbers. Consider a large non-inbred population with mean fitness W. Then, the size of the population reduces to a new smaller value N. In fact, the effective population size should be used here, leading to a progressive increase of inbreeding. Then inbreeding depression occurs at a rate delta, due to partially recessive deleterious alleles that were present at low frequencies at different loci. This means that, in the absence of selection, the expected value for mean fitness after T generations of inbreeding would be. Where is the population mean for Wright's inbreeding coefficient after T generations of inbreeding? 4. However, since selection operates upon fitness, mean fitness should be predicted taking into account both inbreeding and purging, as in the above equation, is the average, purged inbreeding coefficient, after T generations of inbreeding. 1. It depends upon the purging coefficient, D, which represents the deleterious effects that are hidden in heterozygosis but exposed in homozygosis. The average, purged inbreeding coefficient, can be approximated using the recurrent expression. There are also predictive equations to be used with genealogical information. The example of inbreeding depression due to recessive lethals. Average population fitness W against number of generations T of maintenance with population size N equals 10 when, in the original population, each gamete carried on the average one rare recessive lethal. Evolution of W expected only from inbreeding, red line, or from inbreeding and purging, blue line. As an example of genetic purging, consider a large population where there are recessive lethal alleles segregating at very low frequency in many loci, so that each gamete carries on the average one of these alleles. Although about 63% of the gametes carry at least one of these lethal alleles, almost no individual carry two copies of the same lethal. Therefore, since lethals are considered completely recessive, i.e., they are harmless in heterozygosis, they cause almost no deaths. Now assume that population size reduces to a small value, say n equals 10, and remains that small for many generations. As inbreeding increases, the probability of being homozygous for one or more of these lethal alleles also increases, causing fitness to decline. However, as those lethals begin to occur in homozygosis, natural selection begins purging them. The figure to the right gives the expected decline of fitness against the number of generations, taking into account just the increase in inbreeding F, red line, or both inbreeding and purging, blue line, computed using the purged inbreeding coefficient G. This example shows that purging can be very efficient in preventing inbreeding depression. However, for non-lethal deleterious alleles, the efficiency of purging would be smaller, and it can require larger populations to overcome genetic drift. Relevance to the evolution of reproductive systems. Inbreeding depression and purging play a major role in the evolution of reproductive systems. As an example, they determine when selfing becomes at an advantage compared to outcrossing. 5-6. Another example is the genomic renewal in yeasts. Saccharomyces cerevisiae and Saccharomyces paradoxus have a life cycle that alternates between long periods of asexual reproduction as a diploid, ending in meiosis that is usually immediately followed selfing, with only rare outcrossing. 7. Recessive deleterious mutations accumulate during the diploid expansion phase, and are purged during selfing. This purging has been termed genome renewal. 8-9. Evidence and problems. When a previously stable population undergoes inbreeding, if nothing else changes, natural selection should consist mainly of purging. The joint consequences of inbreeding and purging on fitness vary depending on many factors: the previous history of the population, the rate of increase of inbreeding, the harshness of the environment or of the competitive conditions, etc. The effects of purging were first noted by Darwin 10 in plants, and have been detected in laboratory experiments in invertebrate populations undergoing inbreeding in zoos or in the wild, as well as in humans.